InvivoGen has established for more than 40 years, since the 1977, and we have been focusing in innate immunity, providing a wide range of ligands from the very old school LPS poly-IC to new inhibitors and neutralizing antibodies for COVID-19 research. And in Japan, we work with Nacalai test to deliver the best service to you. The presentation will be divided into 3 parts. First part about STING and its current position in tumor treatment. Second part involves inflammasome and the general research direction of how to solve the problems we're now facing. And the last part will be some take-home messages from today's discussion. First, this is the Seagate STING signaling pathway. The presence of cytosolic dsDNA, for example, through DNA virus infection, is recognized by Seagate. Binding of Seagate leads to the formation of Seagate DNA complexes. And this molecular process allows proper activation of the enzymatic function of Seagate, leading to the generation of the 2,3-Cgam, a potent ligand of STING. The Cgam will activate STING, and STING translocates to the Golgi, where it activates downstream signaling to drive the transcription of type 1 interferon and pro-inflammatory genes. Later, it was discovered that STING can be directly activated by CDN, including, more importantly, the artificial ones. So this allows for the direct modulation of this pathway using man-made molecules. The involvement of STING in human diseases has been well documented. This regulation of STING leads to autoimmune diseases like SAVI. And as for now, various residues on STING have been pinpointed as a cause for this dysregulation, paving a path for treatment. Meanwhile, STING is also known to be involved in immune recognition of tumors. Here, I'm showing you the first paper mentioning this involvement. And we can see the knockout of TMEM173, which is STING, causes a decrease in interferon beta production, as well as the response to immunotherapies. Now, if we can see the decrease in STING, the triangle will lead to a higher tumor volume. So this shows STING is involved in the immune recognition of tumors. The role of STING in tumor can be quickly summarized into 2 parts. Firstly, STING activation produces interferon, making the tumor hotter and sensitizes the tumor to immune checkpoint therapies. Secondly, STING activation can promote infiltration of tumor-specific T cells. So this is a very interesting target for drug development. And the drug development against STING has been a great interest for the industry. This timeline outlines the patents and the deals in the industry for you to get an idea of how much effort has been put in. Since 2002, since Aduro Biotech, different companies have joined the conversation, and we have seen some exciting compounds, including the ADU-S100 and a very recent one from Takeda, the TAC-676. Now, here's a summary of the STING agonist in oncology clinical trial. You can look into the individual trials by the NCT numbers, if anything draws your attention. But what I would really like to show you here is the trials are mostly in the early stages. And very recently, we suffered some additional setbacks. 2 big players, Merck and Aduro. First, Merck's compound only had a modest effect in tumor therapy, while for Aduro, Novartis no longer put Aduro's compound in the active pipeline. Basically, Novartis drops the ADU-S100 project. So a lot of new players have joined the hunt for the STING agonist, but it seems that the current agonists are lacking something that we have not fully understood. In the past, the approach from different groups is pretty much the same, including us. By mimicking the natural CGAM, but adding some modifications here and there, we hope to improve the stability and activity of these compounds. So as you can see from here, the compounds developed by different companies, they look highly similar to the natural CGAM. Only some differences in the side groups and the ring compositions. For example, you can see this G here, and for Aduro Biotech, they change it to A, and for us, we change it to I. And then here we also make some changes from this side group. This is natural. This is what Aduro Biotech done, and this is what we have done. The way we found the best side groups and the ring compositions was by reporting cell lines. We generated hundreds of CDNs by making systematic modifications, and then we tested the compound library by reported cell line screening. Here, I'm showing you part of our published results, in which we screen the CDNs, a lot of different compounds, with reported cells of both mice and human background. We assess the compounds by the EC50. On the right, this is basically the same set of results, but with a more visual representations. And now with this, you can clearly see the compounds that stand out. The two with a really low EC50. And so these may be an interesting compounds that worth further discussion. So with this system, we could easily pick up the interesting hits and also had a quantifiable comparison between our compounds. All these cell lines, all these systems that we used are now available for purchase. And the idea of this cell line has been introduced by Dr. Takamatsu. Basically, we overexpress the protein of interest, the sense of interest, for example, STING. And then downstream, we couple it to a Lucia luciferase reporter conjugated to an ISRE transcription site. When this cell senses the ligands, it will activate the ISRE, producing the reporter genes. And the Lucia luciferase is a secretory protein, so we can collect the samples in cell culture supernatant without the need of cell lysis. And after the measurement, we can quantify the effect of the agonist. With this, we hope to help scientists to discover novel and better STING agonists. Recently, GSK developed an agonist that is quite different from what is available so far. Remember, I told you that most compounds are actually modified from natural CGAM. So personally, I'm quite pessimistic about them and believe that they may eventually face the same problem as Merck and Aduro Biotech is facing now. However, this new compound is completely different and may bring some hope. It is completely novel in two ways. First, it is a non-cyclic dinucleotide agonist. And secondly, it allows for intravenous administration to treat tumor. The previous ones, the CGAM similar mimics, can only be used for intratumoral injection. So this one is different. And this compound developed by GSK has demonstrated good effect in tumor model. So this is a tumor model, and you can see the tumor volume, the one that stands out, the red one, is the compound 3, the diapsy, new STING agonist. And for the survival rate, the one that stands out in red is the compound 3 from GSK. So tumor model, it works very well. Later, it was also found to work well with SARS-CoV-2. Again, if we take a look at the survival rate, the two groups that stand out, red and blue, are the diapsy, the new STING agonist. And also, if we take a look at the weight change, the two groups that stand out, red and blue, are the diapsy group with the new STING agonist. So this is a very good compound and very promising. Later, Roche also picked up this molecule and demonstrated that it works both in mice and human system. This is the THP human system. This is the raw mice system. The blue is the diapsy, new compound, and the yellow is the old CGAM. So Roche demonstrated that it worked in both mice and human systems and has a higher potency than natural CGAM. So I believe this will become a new benchmark for drug development. That was about STING. Now let's switch gear and talk about inflammasome. For inflammasome, it's very, very new, and we still have not completely understood the pathway. But basically, the inflammasome is a platform for caspase-1 and IL-1 beta IL-18 activation. It can sense a wide variety of PAMs and DAMs, range from DNA, flagellin to crystals. This regulation of inflammasome has been seen in tumor. For instance, we can detect a clear activation of the IL-1 beta in breast cancer samples compared to surrounding healthy tissues. When we move to mice model, blockade of IL-1 receptor by anakinra, the red group, significantly reduces tumor volume. Similar observations can be seen in Alzheimer's disease. Inflammasome is activated in 80 patients compared to control if we look at the caspase-1 activation. And in mice model, the NLP3 inflammasome inhibitor, MCC950, show a complete restoration of mice cognitive ability. So the APP-PS1 is the Alzheimer's disease model. So you can see the decrease in the cognitive score. And if we add the blocker, it completely restores the cognitive score. So inflammasome itself is a very promising drug target. So far, there are several inflammasome inhibitors marketed for treatment of autoimmune disease, including anakinra and canakinumab from Novartis. They are all marketed to treat autoimmune diseases. Now, the aim now is to transfer this success to treating cancers and neurodegenerative diseases. Several leaders in the field, including Novartis, have been very active and have reported recently their findings from different clinical trials. From Novartis, they report their CANTOS trial, which is a cardiovascular disease clinical trial. In the trial, there is one section that has raised some major concerns. By inhibiting IL-1 beta, you're actually switching off a large area in the immune signaling pathway. So the side effect of this is that it will give you an increased risk of fatal infection. So canakinumab, the IL-1 blocker, is associated with increased risk of fatal infection. So in the worst situation, this may not only apply to canakinumab, but all the other IL-1 blockers as well. So what should we do? If we take a look at the pipelines of the field leaders, we can tell that NLP3, we can see the industry is adapting by looking more upstream. So this is a patent landscape. And you can see the majority of the current patent has moved to NLP3 from the IL-1 receptor. So the industry is looking more upstream. For example, working more on the NLP3 sensor instead of the very downstream IL-1 beta. And from the timeline, you can also see the clinical trials and the deals that it has been a very busy few years. From 2018, a lot of deals has been going on and a lot of clinical trials has been carried out. So it demonstrates the prospect of inflammasome study. Now, if we focus on the field leaders, Inflation, FM Therapeutics, and Northera, they are the field leaders in small molecule development in inflammasome. We can tell that NLP3 inhibitor is the holy grail. They all aim to develop NLP3 inhibitors, no matter it's brain penetrant or systematic peripheral NLP3 inhibitor. This is the holy grail for the industry, NLP3 inhibitor. So of course you may ask, why people focus on NLP3? Other inflammasome sensors like NLP1, NLC4, they are discovered at around the same time as NLP3. And they are also very important and also associated with different diseases. But why people only works on NLP3? I believe one reason why scientists focus on NLP3 is because of the availability of tools and platforms. Compared to others inflammasome sensors, which requires the transfection of intracellular ligands to get it activated, the NLP3 activation system is easier to handle. There's available of simple extracellular activators. You add it in the cells, supernatant, and it works. So this causes the study's bias towards NLP3 and it attracts more resources to NLP3. But I believe later, if we have the better systems for NLP1 and NLC4, these proteins will also catch up to NLP3. Until now, there's no well-established animal model for inflammasome. So we can only rely on cell model for study and screening. One way of doing screening is to use the ASC specs. The ASC is a protein that during inflammasome activation, it will change the localization from evenly distributed in cells to become a condensed dots or specks in the cells. And it can easily observe under microscope. This can be used as a readout for inflammasome activation. And Egles, the founder of IFM Therapeutics, one of the leaders in inflammasome drug development, also mentioned using ASC specs as a readout for inflammasome activation. So here you can see this is without activation. The ASC is like this. With activation, ASC changed localization to really bright dots under microscope. So you can use it to do high throughput screening. And I believe this is the system that IFM Therapeutics is using in-house. However, this method may be more technically challenging to labs that are not familiar with microscopy. So we provide alternative similar to a stain reporter system and similar to the system that Takamatsu Sensei has talked about. We offer the inflammasome reporter cell lines by overexpressing the IL-1 receptor and the reporter gene. So there are two ways you can play around with the system. So first, you may administer different treatments to the cells, for example, your compound library, and then to see the effects. So let's say these three ones are the ones that stand out. So this may be interesting hits that you're looking for. Or you may change the test cells. You may change your experiment background with different mutants or knockout cells to pinpoint action target. So in this experiment, they are studying NLP1. So they use the NLP1 expressing cells. And then for other comparison, they use different NLP1 mutant cells to pinpoint the target. So this is also something you can do. In addition to mutant cells, you can also use knockout to narrow down your point of action. So before I end, I would like to show you two extra screening platforms that we offer. First, a COVID-19 screening platform. The system consists of two cells, a MyDXT expressing cells and a human ACE2 expressing cells. If we overexpress spike protein in these cells, the MyDXT cells will have the spike protein expression. And then if we do a co-culture with this ACE2 cells, the spike and ACE2 will interact and it will cause the two cells to fuse. This result in the transfer of the MyDXT protein from cell 1 to cell 2. The result of this transfer is the activation of the NF-κB and the reporter gene. So with this, the reporter gene can be used to quantify the binding between spike and ACE2. And here we can use this system to screen the effect of different antibodies on different spike proteins variants. So this is Wuhan and this is UK variant. And you can see they work similar. So the currently available neutralizing antibody should work well, at least for these two variants. If unfortunately yet another new variant appear in the world, we can transfect the new spike into these cells and then to study efficacy of current treatments. The second one is the ADCC and ADCP system. So it's not very related to today's talk, so I'm going to go through it very quickly. This is for screening antibody drugs. We replace the use of primary cells to Jurkat cell line. So traditionally, you need to use primary NK cells or macrophages to do this kind of experiment, which is very tedious, and we change it to Jurkat cell line. And also, we use a reporter gene to display the activation of ADCC and ACP. So much simpler than quantifying the real cell death of phagocytosis. So these are some extra platforms or extra tricks you can do with our reporter cell lines. And going back to our main topic, the take-home messages are both STING and inflammasome are potential drug targets. There have been candidate drugs in clinical trials, but they have faced some barriers. And here we hope the reporter cell systems can offer an easy-to-use and efficient platform for novel drug discovery. With this, I will end my presentation. I'm happy to take any questions and comments. Thanks very much for listening.